Good afternoon. Thank you for joining me. My name is Bart de Laat. For the next 30 minutes, I want to talk to you about the results of our recent study on the effect of exercise on the dopaminergic system in individuals with Parkinson's disease. So this presentation consists of 3 parts: the introduction, the methods, and results. And I just want to start with the introduction and see what work has already been done studying the effect of exercise on Parkinson's disease. And we're mainly going to focus on clinical trials here. 2 large ones have been completed that both used long periods of 6 months of intense exercise 3 times a week. And both studies concluded that there was a significant slowing in the motor progression of the disease. So people that exercised, their symptoms seemed to worsen less than those people that did not undergo the same interventions or that did not exercise. Of course, those were very encouraging results, and they spurred actually several other clinical trials that are either recently completed or still ongoing, and evaluate different types of exercise and the effect on different outcomes. So we are certainly not the first people to look at the effect of exercise. Of course, all these studies were mainly looking at clinical symptoms, saying, so the clinical evaluations, how did they progress before and after exercise? What was the effect of exercise? How the positive effect of exercise comes to be, we are not entirely sure. So there is more and more evidence from animal studies, but also from human studies, that suggests that exercise has sort of a combined effect where it suppresses inflammation, where it promotes vascularization. So there's more and there's better blood flow, and at the same time that it increases nerve growth factors, which we can imagine is very beneficial in a neurodegenerative disease. And so I think the most common hypothesis currently is that these 3 factors are actually combined responsible for the neuroprotective effect of exercise that we see, or at least suggest happens on the dopaminergic neuron. You might have seen this image quite a few times in the past few days, but I just want to go over it once more. Here in the small pink region is a substantia nigra, which is where the cell bodies of the dopamine neurons lie. And we can see that they branch out into the different regions of the striatum, being the caudate and the putamen. And if we take a closer look at the substantia nigra, which is literally black substance, because it contains this melanin, much like the melanin in our skin, but here it's produced by neurons, so it's called neuromelanin. And as we zoom in closer, which is done in these insets, we can see that there's neurons, dopaminergic synapses, where dopamine neurons actually come out. So you can see that there's dopamine coming down from this first neuron, which is being released, so it can actually trigger a signal through the dopamine receptors in the second neuron. The dopamine that's still floating around in this area that we call the synaptic cleft is pumped back into the neurons by dopamine transporters to avoid that there is a sort of a constant low amplitude signal. This is the situation in a healthy person And schematically, what it would look like in somebody with Parkinson's disease is like this. So there's less dopamine. There are fewer dopaminergic receptors, and there's also fewer dopamine transporters. Why? Because they are upregulated if there's a lot of dopamine floating around, and just that needs to be pumped back in. Whereas here, of course, we don't have that much dopamine anymore, so there's less need for them, and therefore they are downregulated. So this slide kind of summarizes the two outcomes of our study, which is on the one hand neuromelanin, and on the other hand the dopamine transporters. So that brings us to the methods. I want to start with the design of the study, where you can see that it's centerpiece really is this block of exercise. So it's a 6-month exercise block, like the early clinical trials, where people are asked to complete at least 3 sessions per week and aim to reach 80% of their maximum heart rate. That's the definition of intense exercise, and that's what we know would have caused these clinical benefits. In addition, we asked participants to complete a weekly survey, both on their experience and the satisfaction with their program. So how are they experiencing the classes? Is it intense? Is it too much? Are they happy with how they're performing? Are they happy with the trainers? Are they experiencing any difficulties? Things like that. Before and after those 6 months, people would come in and they would undergo both clinical evaluation and 2 neuroimaging techniques, which we will come to a little bit later on. For us, this first period was very important as well. So that's a trial period where people were asked to do the exercise program for 2 weeks. So that was to evaluate if people were really up to such a strenuous and intense exercise program, and if they thought they would be motivated to complete it for 6 months, and thus avoiding the situation where they would come in, be scanned, and then during that 6 months would actually say, no, this is a bit too much for me, and then basically we wouldn't have their before and after data. The exercise program that we used is called Beat Parkinson's Today, and it was local to us in Connecticut, where also Yale University is. And much, I think, like PD Warrior, it's evidence-based exercise program for people with PD that gives guided training sessions, both online and in person. And they do a great job and aiming for maximal intensity, but at the same time also adjusting their trainings to everybody's individual capabilities and restrictions. So these are just a few screenshots of different exercises that were performed, where you can see that it's loosely based on a boxing class, and there is often a circuit where people would do multiple exercises, and then every month there is a focus. For example, there's balance month, where there's a concentration on those specific skills and those exercises that align with that focus. For us, it was really important that people would reach their target heart rate, so that 80% of their maximal heart rate, and to make sure that they did, or at least to evaluate that objectively, we would equip everybody with a chest strap. So that's a heart rate monitor that would sync with their phones, and therefore we could see their data online. This is an example of that. So you can see that over time, their heart rate rises during the warm-up. We can see that it's the high intensity interval training program. So people are allowed to rest and the heart rate comes down a little bit, and then they go back in and peak. The red dashed line is the threshold here, or it's their aim, whereas the green line is the average of the heart rate between 20 and 50 minutes, which is sort of the typically the intense period of the class. You can see that this individual did really well for this specific exercise session. That brings us to the neuroimaging technique. So there's two that we used. One is positron emission tomography, or PET, and the second one is magnetic resonance imaging, or MRI. So we're going to start with PET, which uses radioactive tracers. Those are molecules that consist of two parts. One is the radioactive part, whereas they're bound to a molecule that will bind to a biological target. What that means is if we inject it, it will circulate through the body, but it will accumulate there where there's most of the target. And as the radioactivity is decaying, it will send out a signal which is detected by this ring of detectors, and thus giving us the signal that we need. Now, the tracer that we used was specifically made so that it would bind to these dopamine transporters. That means that the higher signal we would observe, the higher concentration or more dopamine transporters will actually be present in the brain. So these, and then the top row are PET images. This is a bit what it would look like. So you can see that in those regions where this is striatum, where there's a lot of dopamine transporters, we see a lot of signal, right? So that the colors turn greenish, yellow, and even red. Whereas in the rest of the brain, there are very few to none dopamine transporters. And we're more in the blue, purple, black colors. We can see the substantia nigra lighting up a little bit. So that's that for PET. The MRI that we used is a standard magnetic resonance imaging machine, which uses strong magnetic fields, not radiation. And if programmed correctly, it can actually measure neuromelanin. Ironically, substantia nigra, because of the magnetic properties of the neuromelanin, lights up as a more white region on these images. And for example, here we can see the white substantia nigra, which gives us an intense white signal that is in healthy people if there's a lot of neuromelanin. This is an example of somebody with PD, where most of the neuromelanin is lost, and we can see that brightness sort of fades into the same intensity as the background region. Okay, so I think we're ready to dive into the results. In the end, 10 people completed our study. Everybody had mild disease, was within 2 years of diagnosis and around 65 years old. We saw that movement exam scores were essentially unchanged after 6 months, so that's good. And then also some improvement in time motor functions and also in quality of life scores. The survey results, as you might remember, people filled out a survey once a week, basically summarized the whole experience as intense, but very beneficial, well tolerated. We did see some side effects like muscle soreness, knee pain, back pain, which I think are typical for exercise in general and particularly in an older cohort. These are the results from the PET. So the PET images that you see is the average of the entire group. You can see that the signal in the striatum, it hasn't changed that much, but particularly in the substantia nigra, which is in the cutouts here, there's a strong increase in after exercise compared to before. So that suggests that there is indeed more dopamine transport after exercise. These are the same data, but it's a different way of showing them. So each line is an individual participant. Those that increased after exercise are colored red. Person that decreased after exercise is colored blue. And the thick black line shows the average increase, the average difference, but it is an increase of our cohort, so of the people that participated in our study. And the dashed line represents the normal decrease. So over 6 months, the concentration in dopamine transporters was expected to go down this much, right? So that has been assessed in a different group with many people with exactly the same dopamine transporter PET as we used. You can see that it's quite a stark difference between those two black lines. Similarly, if we look at the neuromelanin MRI results, so this is raw average results, we see that the intensity of the substantia nigra, which we now know is correlated with the neuromelanin presence, seems to have come back quite a bit after exercise. Below is the same image, but here it's expressed as a ratio between background without neuromelanin, excuse me, and substantia nigra. This is the same type of graph as we saw for the PET. We can see that there's more blue lines, so more people decreased neuromelanin content despite the exercise program. But we also still see that on average, there is an increase where we expected a steep decrease, which has been reported for 6 months. So in conclusion, we saw that high intensity exercise may have neuroprotective effects, especially on the dopaminergic neurons in PD. At the same time, I think it's important to realize that more research is needed. We need more research to show that these effects are true in different groups, particularly larger groups of people. I think more research is needed to compare different exercise regimens, intensities, to see how can we maximize this effect, right? How can we sort of optimize our exercise programs to have the most and the best neuroprotective effects. And then finally, and I think that is, for researchers at least, it's sort of the holy grail, is how does this work, right? What is the mechanism? How what's the molecular mechanism underlying these beneficial effects? Because then we might think about emulating that and also amplifying that effect. I am part of a dream team. These were the two PIs of the study, Dr. Tenaz and Dr. Morris. We are, of course, very grateful to Michelle Hespler, who was the founder and the leader of PD Today. Of course, all the rest of our team, and there are actually many more people that contributed their technical expertise to make all of this possible. As you can see on this image, it is afternoon now in Australia, but it is deep in the night in Europe where I am. So I'm unfortunately not available right now to answer your questions live. However, please send them via the chat because the organization will make sure that me and the team will get them. We will make sure to respond to all of your questions and get them back to you. So thank you for listening, for joining me, and good day.